Natural killer cells are activated by interferons and macrophage-derived cytokines to serve as an early defense against certain intracellular infections. Natural killer cells, NK cells, develop in the bone marrow from the common lymphoid progenitor cell and circulate in the blood. They are larger than T and B lymphocytes, have distinctive cytoplasmic granules, and are functionally identified by their ability to kill certain lymphoid tumor cell lines in vitro without the need for prior immunization or activation. The mechanism of NK cell killing is the same as that used by the cytotoxic T cells generated in an adaptive immune response. Cytotoxic granules are released onto the surface of the bound target cell, and the effector proteins they contain penetrate the cell membrane and induce programmed cell death. However, NK cell killing is triggered by invariant receptors, and their known function in host defense is in the early phases of infection with several intracellular pathogens, particularly herpes viruses, the protozoan parasite Leishmania, and the bacterium Listeria monocytogenes. NK cells are activated in response to interferons or macrophage-derived cytokines. Although NK cells that can kill sensitive targets can be isolated from uninfected individuals, this activity is increased by between 20-fold and 100-fold when NK cells are exposed to interferon alpha and interferon beta, or to the NK cell activating factor IL-12, which is one of the cytokines produced early in many infections. Activated NK cells serve to contain virus infections, while adaptive immune response generates antigen-specific cytotoxic T cells that can clear the infection. At present, the only clue to the physiological function of NK cells in humans comes from a rare patient deficient in NK cells who proved highly susceptible to early phases of herpes virus infection. IL-12, in synergy with TNF-alpha, can also elicit the production of large amounts of interferon lambda by NK cells, and this secreted interferon lambda is crucial in controlling some infections before T cells have been activated to produce this cytokine. One example is the response to Listeria monocytogenes. Mice that lack T and B lymphocytes are initially quite resistant to this pathogen. However, antibody-mediated depletion of NK cells or neutralization of TNF-alpha or interferon lambda or their receptors renders these mice highly susceptible, so that they die a few days after the infection before an adaptive immune response can be mounted. And that's natural killer cells are activated by interferons and macrophage-derived cytokines to serve as an early defense against certain intracellular infections. Have a great day.